good evening to everybody. Thank you all so much for coming along tonight for tonight's event, which is part of Scotland's Digital Science Festival powered by TechFest. I am now delighted to introduce you to tonight's speaker, Daniel Berg. Daniel is very much back by popular demand. He's a researcher at the University of Aberdeen, and whenever possible, we try and convince him to come along and be part of whatever TechFest is doing because his research in neuroscience is just so fascinating. In fact, Daniel's talk on how to grow new brain cells is one of the most popular videos on our YouTube, which we're really delighted about. So if you haven't seen that, you can go on over to our YouTube channel and have a watch of that at some point. And we were delighted to convince Daniel to come back for tonight's talk because we're going to be learning about the human brain and learning about how it develops. So it's not quite a sequel, but it's almost a continuation of the presentations that Daniel's done for us in the past. So thank you so much for your time tonight, Daniel, and I will go ahead and turn off my camera and pass over to yourself.
Great. Thank you so much for that kind introduction. And thank you to TechFest. It's great to have this opportunity to learn lots about lots of different science and what's going on in the different fields. So as you heard, my interest is in the brain and the human brain in particular, because I like to say I like to think that the human brain is the pinnacle of evolution. Now, other scientists are going to disagree with me on that, but the human brain is truly remarkable in what it can do. So if we look at some of my favorite things that the human brain has been able to done, it's been able to compose lots and lots of beautiful music. It's done amazing art with paintings, and it's amazing stories. I just took a picture of a library just to illustrate all the amazing books that the human brain has been able to invent and write down. So the human brain is truly remarkable. And we're also able to do science. We're able to do medical science that we're going to talk about today, and physical science. I've just chosen a few here. So don't be offended if I've left out your favorite art or your favorite science. But these are the just to illustrate how amazing the human brain is. So, what makes the human brain different from other brains, what from other animals? So, here's an illustration of the human brain. Hopefully you've all recognized it. We have this big cortex here with lots of folds. We have the cerebellum here at the back, and then down here we have the spinal cord going down on the brain stem. If we compare the human brain to brains of other animals, other mammals actually, it's not extremely remarkable, the human brain. It's not the biggest brain. If we look at this whale brain here and also the dolphin down here, they are both bigger than the human brain, and they have more neurons in total. So that doesn't make the human brain special. But there is one region of the human brain that is special, and that's this cortex here. This big cortex contains much more neurons compared to any other cortex if you look at lots of animals. So neurons, I mentioned neurons. I'm just going to go through here what neurons are. So neurons are the cells in the brain talking to each other. And right now in your brain, they're sending signals and they're relaying the information from your ears. You're hearing my voice. And there are lots and lots of neurons, and they come in different shapes and sizes. So I've just illustrated this is what a unipolar neuron. It's got the cell body here and sending out the axon. Here's a bipolar neuron. You can see it's got 2 feet here. Here's a pseudo unipolar neuron, and then we have the multipolar neuron. So these neurons come in lots of different kinds. They express different proteins and they have different functions in the brain. And in the human brain, we have about 86 billion neurons. This is you see, of course, it's difficult to count neurons. Yeah, but this is the current estimate is that it's about 86 billion neurons. And each one of these neurons has on average 7,000 synaptic connections. So they're talking to each other in 7,000 synaptic connections, which means that together in the brain, we have about 100 trillion. I'm pretty sure this is a trillion. I counted these zeros before, and we have about 100 trillion synapses in the brain. So these are these places where the neurons are talking to each other, sending synapses and sending signals to each other. So the amount of neurons in the cortex, this is what makes the human brain so special, and that we can create all this amazing art, and we can have these thoughts that we're having right at this very moment. But these neurons aren't the only cells in the brain. They're usually the superstars. You always talk about neurons, but there are lots and lots of other cell types in the brain. And these are, for example, we have these astrocytes. So these are glia, what we call glial cells. Sometimes a bit derogatory called the neurons helper cells. They help the neurons, but we've discovered recently that these cells are very important for the brain, the neurons to survive. They clean up a lot of mess and even send signals to the neurons. In the brain, we have about 86, we have the same amount, about the same number of astrocytes as we have neurons. So 86 billion astrocytes in the human brain. Another cell type is the oligodendrocytes. So these are very, very important because each arm here wraps one part of a neuron and make sure that the signal can go really fast. So these oligodendrocytes are important for signals to go properly in the neurons. And we have about 50 billion of these cells. And there are some diseases when these cells die, and that leads to a lot of trouble in the brain. Another very important cell types are the microglia, and we have about 10 billion of these cells. Microglia are the brain's immune cells. We don't have the same immune cells in the brain as we have in the other parts of the body because the brain is especially sensitive and we need to keep it very clean. But these microglia go around uh, seeing if they can find any waste products or any dying neuron, then they degrade those cells and eat them up. And those are the microglia. And then finally, we also have blood. We have vasculature. We need to have the blood circulating in our brain. And it's been estimated that if you take all the vasculature in the brain and put it in one long line, it's about 650 kilometers long. So if my computer is correct, 650 kilometers is about the distance as the bird flies from Aberdeen down to London. So that would be all the vasculature in one long row. So lots and lots of vasculature in the brain. And this vascular system also contains lots of different cell types. You don't have to remember these cell types. It's not going to come on the test, but we have the endothelial cells here, which are part of the blood vessels, but also cells supporting here and also making sure no bad stuff that's in the blood comes into the brain. So we have an important barrier here between the blood and the brain because things that we eat, if we sometimes eat something bad, we don't want that stuff getting into the brain. So we have lots of important cell types here. And for a normal brain to work no, well and everything to work normally so that we can create this beautiful art in the Scottish National Gallery here by William McTaggart. All these cells need to be healthy and need to be communicating with each other in a good way. So the neurons, of course, the neurons need to fire to each other with these synapses here, but the oligodendrocytes also need to insulate these neurons. The microglia need to make sure there's no bad toxic stuff in the brain. We need to have the blood vessels here, and we need the astrocytes. So all these cells need to help each other and be healthy for the brain to function normally. And as you all know, sometimes the brain doesn't work normally, and this can be illustrated quite nicely looking at some artists' work. So Louis Wayne was a painter, and he loved cats like many other famous artists. He loved cats, and he drew lots of paintings of cats. And it's people have argued a bit what happened to his brain, but some claim that he had schizophrenia or he had some brain damage, but he probably had some mental condition, and that could be very clearly seen in his the way he painted his cats. Sometimes he painted beautiful kittens, very realistic, but then sometimes you could kind of sense that he was going through some really hard times, and they look very psychedelic cats. So this is just to illustrate how artists you can get the picture of the artist's mental health through their art. Another example here is William Uttermohlen. I'm not sure how to pronounce his name, but he was from Philadelphia in Pennsylvania. And then in 95, he was diagnosed with Alzheimer's disease. So Alzheimer's disease, as you know, is a disease when neurons in the brain slowly die. And these uh, patients, they will slowly start having worse memory, but they also have other effects like depression. And William kept painting self-portraits as his disease progressed. And when you look at his painting, you can see that the way he sees himself changes. So he's losing his spatial awareness, and the details of the paintings are changing. Also, he becomes more abstract and it changes in color. And when, for example, these paintings where eyes and things are in the wrong place, when people would ask him, "Can you see that this painting is wrong?" He couldn't see it himself. So with Alzheimer's disease, he lost some spatial awareness and detail. So what actually happens in the brain in these disorders? So I've talked firstly about we have neurodevelopmental disorders. So that's schizophrenia or other people on this other neurodevelopmental disorders. That's when something goes wrong during development. And then we have dementia like Alzheimer's disease. As you can see here in this is the Alzheimer's disease brain, and this is a healthy brain. You can see here it's much thinner here. Many neurons have died, and it's actually the cells in the brain that are dying in Alzheimer's disease. With these neurodevelopmental disorders, on the other hand, it's different things. If this is a normal brain, microcephaly here, this is a much smaller brain. So that's a neurodevelopmental disorder. We also have lissencephaly. As I said before, the human brain is very got lots of folds here, but the lissencephalic brain is completely flat. And then we also have other disorder like focal cortical dysplasia, when there are some um, problems with the folds. So these are some examples on uh, disorders where something goes wrong in the human brain. And as a neuroscientist, I want to find what's the cause of these diseases, and how can we cure them, or how can we prevent them, or just what's happening in these cases. And traditionally, when people study neuroscience, people have used animal models. For example, the mouse brain here. In the lab, many people use mice because we can do experiments on them, and it's not so controversial. But as you can see here, this is relatively to scale. You can see that, of course, the mouse brain is very much smaller than the human brain. But there's also another difference here, and I'll illustrate that by blowing up the mouse brain here. Uh, the mouse brain is flat, while the human brain has these folds. So there are very many differences, not only in these big differences like the size and the folding, but actually each one of these cell types that I've told you about today are a bit different in the mouse and in the human compared to the human. So if we really want to learn about how the human brain works and what's the problem in the human brain in these diseases and disorders, we need to have a way to study the human brain. So how can we do that? How can we study the human brain? If I ask you, can I take a few neurons from your brain and grow them in my lab? Hopefully you're going to say no. I don't want to give up my neurons because that's not very good. So that's not a good way. We can take neurons from deceased people if they've given consent, but they are always going to be not very healthy neurons. So luckily, scientists have developed a way that we can generate neurons in the lab. We can grow them from a dish, in the dish. And the way that we do that is that we take skin cells from patients or normal people. I can take your skin cells and then I'll grow them in the lab in a little petri dish. And then, thanks to this guy, Shinya Yamanaka, he discovered that if he overexpresses, if he takes these signaling molecules or these chemicals and put them on on the cells, on these skin cells, they're gonna revert and become stem cells. So then I can look at if I've taken your skin cells, grown them, and then added these proteins to them, I then have your stem cells in the dish. And stem cells are remarkable cells because stem cells can become anything in the body. So if I have your stem cells, that means that I can put other chemicals on those cells, and then I can get your brain cells in the dish. Look at some of brain cells with your genetic background in the dish. I can also look at muscle cells and immune cells. So this is really a powerful tool. Here in the picture here, I'm showing you some neurons that have been stained with different proteins, and you can see that they're communicating with each other. They're sending out projections, and this is really great because then you can take um, neurons from skin cell from someone who has schizophrenia and someone who doesn't, and then you can compare those neurons and see what's the difference. But these are all attached to these cell plates. And that's not really ideal because in these dishes, we mostly only have one cell type per dish. That's the way these methods to generating these cell types work. So mostly just one type of neuron here or one type of muscle cell or one type of immune cell. You can mix and match, but it's usually one per um, per one cell type per experiment. Also, these cultures are two-dimensional. They always attach to the plastic bottom here of these petri dishes, and the human body is not two-dimensional. It's three-dimensional. And also, these cells don't have the shape that they would normally have in a three-dimensional body. So to try and circumvent this, people developed what's the main topic of this talk today, and that's brain organoids. So they discovered that if we have these stem cells in the dish and then we make them into little clumps and make sure that they stick together, then grow them in this what's called like a cell incubator. And this is a stirrer here. So this bar here is going to mix these ups just to make sure that they don't attach to the bottom like they do here. And then you give them the right signaling molecules to make sure that to try and make them into brain cells. And then these cells are going to be called what we call brain organoids. So these are going to become 3D structures with neural stem cells and neurons in these little clumps. So it turned out that this cell incubator here, we need to add all these chemicals and this growth media that we add here, the red stuff here. It's very, very expensive. And this is quite a big bottle. So it turned out that if we can just grow them in these wells here and then put them on a shaker and then put this shaker in an incubator. That's a much cheaper and just as an efficient way to grow these organoids. So this is a way to generate brain organoids, growing from stem cells, making the clumps, giving the cells the right molecules to make brain cells, and then we get organoids. And here are some examples of these organoids. So one thing you can see on this picture here is that it takes quite a long time for them to grow in the lab. So this is day 8, day 14. You can see them growing to day 25. But if you want to really get neurons and them starting to connect with each other, you'll have to wait until about day 80. And it it's very these experiments are very expensive because we need to feed these cells every day. So my students who are doing these experiments, they have to come in every day to feed these organoids and make sure that they're happy. And these organoids don't care if it's weekend or Christmas, they have to be fed every day. And I said that these organoids are three-dimensional. So here we look at cross sections of these organoids, and you can see that there are different cell types here. For example, we have let's look at this one. We have the red here. The red is the stem cells in the center. And then here we have some TBR2, that's the blue. And then outside we have neurons in green. So we have stem cells and these intermediate progenitor cells in blue, and then we have the neurons in green. So we have multiple stem cells, multiple cell types here in one organoid, unlike in the two-dimensional culture where we only have one cell type. So this is very similar to the developing human brain. Um, we can yeah, these are just looking at the neurons here. So these organoids are very good tool to study the early development of the human brain. So I'm going to give you some examples of how these organoids have been used in the last 10 years. Some of you might remember that in 2015-16 there was the Zika virus pandemic, mostly in Brazil and in South America. And this was a virus that it was spread by mosquitoes, and some of the pregnant women who got bitten by a mosquito and got Zika virus, their babies developed smaller heads and their brain was much smaller. So this is called microcephaly. This is what I showed you one of these brains before. And these children will have intellectually impaired. So this was a very scary, of course, but we didn't know how the Zika virus could do this, how it could result in babies with smaller brains. How does the virus do this? So what people did is that they had these brain organoids, and then they added Zika virus to the organoids, and then they could see the green here is this where the Zika virus has infected. And they could see that the Zika virus actually infected the neural stem cells, the stem cells in the brain. And here you can see these are dying cells. So it led to many, many more dying neural stem cells in the brain. And that's the way that the Zika virus can kill neural stem cells and in the end result in a smaller brain of these patients. And a more recent pandemic, of course, is the SARS-CoV. And you, some people who have been with SARS infected with SARS-CoV had long COVID, and some have problems with the brain. So people wanted to study, does this virus infect the brain, especially during development? And here was a study by Fadi Jacob, and he couldn't actually see many uh, infections in neurons or neural stem cells or the glia cells. So these cells don't get very much infected by the SARS-CoV. But it turns out another cell type in the brain is infected, and this is the choroid plexus. So this is an organ in the brain that produces all the fluid that's inside the brain and around the brain. It's really important for the brain to be healthy and get the right nutrients. And these holes in the brain are filled with cerebrospinal fluid. So the choroid plexus produces this fluid, and it can be highly infected by the, the COVID virus. And now people are trying to find out, could this be a reason why some patients have brain fog or things like this after even long times after they've been infected. So here are some ways that organoids can be used to study neurodevelopmental, especially um, how environmental challenges like virus, how this can affect the brain development. And organoids can be really, really useful for this. And if we look at Alzheimer's disease, people also use organoids to study Alzheimer's disease. So previously I talked about external factor. We can get stem cells from patients who have Alzheimer's disease, and then we can grow their brains in the dish and compare it to a healthy control. And Unche Kang, here at the University of Aberdeen, she's a real expert in making these organoids, and I'm very lucky to be collaborating with her. And she's making these organoids from Alzheimer's disease patients and healthy. And here we can see plaques and tangles. So these are a few of the hallmarks for Alzheimer's disease. And already in these, this is the Alzheimer's disease organoid, and this is the healthy organoid. And as you can see, there's more of this green stuff here in the Alzheimer's disease neuron. And then we can use these organoids to determine how does these green stuff, how does that affect the stem cells or the neurons in these organoids, and then we can try and treat that with some drugs and try to find some signaling how they're communicating, and hopefully in the long term we'll be able to find a drug for these disorders. So most of the organoids that I've been showing you now is from the cortex. So this big curved structure here on the top. But we can also generate organoids for other brain regions. And Olivia, my student, she is trying to make hippocampal neurons. So hippocampus is another part of the brain, and it's really important for learning and memory. And it's actually one of the areas of the brain that is first affected by Alzheimer's disease in many cases. So she's been able to generate these hippocampal neurons. So the red, you can see hippocampal neurons that we can see that they are different from the cortical organoids. She's also been able to take a movie here where you can see how the neurons are signaling. So they're actually talking to each other in these organoids and sending messages to each other. It's they're very primitive, but the neurons are sending electronic signals to each other just as in development. So we can make brain region specific organoids, like Olivia here, who has made these hippocampal organoids. People have made cortex, is what I've been talking most about, hippocampus, like Olivia. People have made midbrain and cerebellum and spinal cord. But people want to try and model when these brain regions communicate with each other. So they want to, it's important how the different brain regions talk to each other in our brain, and that can go wrong in this disease. So the way people have tried to do that is you make two different organoids, one for one brain region and one for another brain region. And here they've colored one red, one brain region, and the green one is another brain region. And then they put them together. And as you can see here, here's day 17 of the experiment, but they have to wait to day 60. So they have to wait quite long, but then they eventually they fuse and they're going to start sending projections into each other. And you can study how these projections go into each other, and you can examine which genes are responsible for certain things, and also if we can if this is disrupted by any environmental factors like alcohol or pollution or and things like that. So these, when you assemble combine organoids, these are called assembloids. And here is another example of these assembloids, when they have assembled different organoids to make a, a more complex brain structure. So here they've got a part of the visual cortex and then the thalamus and then a retinal organoid. So this is how the brain communicates to the eye. And of course, these pathways are broken down in many disorders of the eye. So this information that we can gain from this can be very important to help people in their old age when they start getting bad eyesight, how these axons are sent from the brain to the eye. And these are assembloids. I have another picture here of an assembloid where you can see the red really sending in projections here into this other neural organoid here. All these cells in the organoid come from neural stem cells. And neural stem cells are the stem cells in the brain that can give rise to neurons, astrocytes, and also oligodendrocytes. But as I said in my introductions, these are not the only cell types in the brain. We also have microglia, the immune cells of the brain. And these immune cells are really important for in Alzheimer's disease. We have a lot of work to do in Alzheimer's disease, and they're very important for Alzheimer's disease. And also the blood vasculature. We need vasculature. People have seen that the vasculature is affected in different disorders, and they are not present in these standard organoids that I've been talking about. So we need to try to get these cells into our organoid as well. So one way, again, in Kang, together with our student Mohammed, we've been trying to get, we have the stem cells, and then we have to make both the organoid and then the microglia, these immune cells. We have to make them separately, but then in the end, we can add the microglia to the organoid to try and get them to infiltrate the organoid. And if these green cells here are the microglia, these immune cells in the organoid, busy eating up lots of debris and taking out the garbage. So these are also called assembloids. Assembloids can be putting two organoids together, but it can also be when putting another cell type onto an organoid. A problem with these organoids is that they like to grow, but because that they don't have a vascular system, the center of the organoid is not going to get enough nutrition. It's not going to get the food and the oxygen that they need. So it's going to die. In the center of the organoid, we have lots of cells that are dying, and that's not great for our experiments. And partly the reason is because they don't have vascular system. So we are also trying to get the vascularized organoids. And here you can, this is just a vascular organoid. There's no brain here, but this is just how you can see the vascular system in this organoid. And what people have done in this study here is they've had two of these vascular organoids and then put it around the brain organoid. And then these blood vessels are going to infiltrate this organoid. And these green cells here are the vascular cells in the organoid. So this is how people in the field are trying to solve these problems and trying to get organoids that really look like the human brain in a small miniaturized model that we can do experiments on. But as you can imagine, the field has still a long way to go. We need to these vascularized organoids don't contain microglia, and we need to try and get all cell types in there. And just to summarize what I've talked about today, so the human brain is truly remarkable. It's the great one of the great works of of evolution, and we have all these cell types communicating with each other. And for us to be healthy and have a healthy brain, these cell types need to be healthy, and sometimes they're not healthy. Like when in Alzheimer's disease or Parkinson's disease, some of these cell types die, and we actually see effects on all of these cell types. And if we want to learn what's actually wrong in these disease, we need to have a human model system. And that's what we're trying to do with these organoids. We're trying to see how these diseases actually look in the human, and then we can do experiments on these organoids to try and rescue them and try and find cures for either neurodevelopmental disorders or these neurodegenerative disorders. So that's what we're trying to do in our labs. And with that, of course, I just some of this is my lab. We have, as you can imagine, all of this research is very, very expensive. So we're lucky to have lots of great funders who support our research. And we're also very proud to get from private donors who want to either have family with Alzheimer's disease. Some here in Aberdeen have been very supportive, and we're very, very grateful for that. And with that, thank you very much, and I'll be happy to answer any questions.